Great. Hello, everyone. Welcome on today's webinar on practical virtual screening with CHEESE Search. And I would like to introduce you to Christoph Stach, who is our application scientist with background in medicinal chemistry, and he will present use cases of CHEESE in virtual screening and our recent successes in multiple campaigns we did. Christoph, it's yours.
So, thank you, Miroslav, for introduction. I also welcome you all to our seminar. Thank you all for coming. As the topic of our webinar suggests, we are going to focus on the practical usage of our CHEESE search in virtual screening. And in the beginning, I would like to start with pointing out of two main topics which I would like to focus on. The first is the virtual screening via expansion of library of 22 known inhibitors, which we use in the case of the discovery of novel ligands of BRD4-BD1 active site, where we were able to, with a great success, to expand very limited set of small molecules into wide library in a matter of seconds, while maintaining the main physicochemical properties and biological activity, and also very richly expand the structural diversity of the set, which led into introduction of new compounds and most importantly new structural features that were previously unknown in the set of, at least in the literature, described inhibitors. Another use case we would like to discuss today is our machine learning approach for the binder discovery using our technique, which we applied on DYRK1B kinase in the recent 9th Advanced Drug Discovery Challenge, where we were given a blind set of compounds and a set of verified binders, and only knowing being aware of these compounds, we were able to find hidden molecules in the blind set, while respecting or with the emphasis of the molecular electrostatics, structural shape, and small subtle structural details, which are unknown or invisible using different classical techniques. And thus, we performed very quickly, very fast and quick 3D similarity screening. So, if I will somehow wrap up the topics of today's seminar, we would like to focus on the rapid hit expansion using our CHEESE search technology, which allows us to search ultra large libraries counting around 40 billion compounds within a very short time frame of seconds, and thus implement them into the very straightforward pipeline very quickly, which results into gain of very diverse set of potential compounds that are all enumerated and are very easily purchasable, since our CHEESE search is directly linked to the various vendors. Also, regarding the 3D similarity screening, which I mentioned on the previous slide, we are going to explore the CHEESE Embeddings, which is our technology of molecule description from the point of electrostatics, shapes, and also Morgan fingerprints that are also encoded in these embeddings. And based on these embeddings, we are able to see structural features or patterns which are otherwise invisible from the perspective of classical Morgan fingerprints approaches or fingerprints-based approaches that are classical in our field. And these CHEESE embeddings can be used very quickly with high speed and very high precision. And in the very end of our presentation, I will show you a small sneak peek of our most recent technology, SynthonGPT, which is generative model that can be easily implemented into iterative search for novel binders. But back to the BRD4 BD1 selective inhibitor selection. I will perhaps introduce you the BRD4 protein, which is key enzymatic regulator involved in a transcriptional control of the body and can be somehow exploited by cancer pathways and thus control the uncontrolled proliferation, or thus drive the uncontrolled proliferation of cancer cells. And actually, this is very key factor in the treatment of certain types of cancer, as it has a very strong impact on the cancer cell growth. This protein has two active sites. One is BD1, which is the crucial for this project. It's very narrow, very conserved, and very rigid binding pocket here in the region of the in the crucial region of the protein, and it actually is responsible for many processes regarding the cancer cell proliferation. And then there is also the BD2 pocket, which is not connected with the cancer-related processes. It's more connected to the inflammatory processes. So generally, when we are trying to aim BRD4 protein, we are aiming on the BD1, which is very difficult to select. It's a very difficultly druggable target, and for that reason, we selected this as a showcase of the capabilities of our cheese, since it can expand very easily known library of inhibitors. In this case, it counted 22 inhibitors that were really proved selective towards the BD1 pocket. And using CHEESE search, we expanded this small library to 4.5 million compounds that shared 70% electrostatic similarity with those starting hits. We used all of our databases, or all databases from all vendors within the CHEESE search, and by applying these presets, mainly 70% electrostatics, which was actually used during the user via the user interface on our web pages, cheese.deepmedchem.com. We were able to expand the library to 4.5 millions, and then by clustering techniques outside of CHEESE, we were able to find 500,000 of the most interesting compounds, of the most unique compounds in this set. And then via docking studies, we were able to find around 400 very potent molecules, which were then evaluated and applied back to the clustering procedure, where we did some sort of back enrichment in order to find the most similar compounds from the starting set to those 400, and repeated the whole set. And thus we were able to iteratively gain the most interesting compounds with really high affinity, in silico affinity towards the BRD4 BD1 pocket. As you can see here, we were able to find 12 new candidates in a matter of a few hours, since the CHEESE search is generally very quick. The search within the CHEESE search takes around 3 to 10 seconds, very fast process. And then the most time-consuming way is actually the post-process of the gain results. As you can see here, we were able to introduce novel structural novelties to the starting set of compounds. As you can see, the average Tanimoto similarity when we are comparing those 22 starting ligands against our 12 selected candidates was 0.12. The physicochemical profile was more or less maintained, as it does not has not changed much, or more or less not at all. And as you can see on this PCA plot, also the chemical space that has been occupied by those 22 starting ligands was kept, and we stayed within this same range of chemical properties. as you can, which means that in a very short time span we were able to expand the library of our known hits and gain very interesting compounds based on the 70% electrostatic similarity, which in this case means that it does not necessarily follow the structural pattern. It more or less follows the distribution of the partial charges in the 3D space, which then it's which is what the electrostatic search in our case does. So you can see examples of compounds here in the first column. Those were examples of those starting 22 hits, and in the column 2 and 3 you can see the closest examples of those compounds from our candidate compound. So we can see there is a quite structural diversity in the set of those candidate compounds that we were able to find very quickly using CHEESE search. Now I will continue with the DYRK1B virtual screening challenge, which happened, they took place in the beginning of February, and which we won. Our team was the first amongst 50 another team from all over the world. It was international competition, and we were screening compounds against the kinase DYRK1B, and we were given the blind set with more than 3,000 molecules. And our goal was to find find a number of find active molecules that were mixed in this blind set. We were provided the X-ray structure and a set of verified ligands, set of verified compounds, both active, which there's more than 700 active compounds and more than 1100 inactive compounds. And based on this knowledge, we were supposed to discover a way how to, in a very short time, because this competition took only about 5 hours, in a very short time, develop a model which would be very confident in the selection of the best inhibitors of this kinase. It is important to say that in that particular case, all docking techniques failed, since the binding pocket of this enzyme is very narrow. There is not much space for the for any movement of the molecule. So we really needed to be thorough in the selection of the technique for the prediction of the compounds ability to bind there. As I said, more or less every single team that has tried docking studies or docking protocol on this enzyme failed. So it was necessary to actually discover some another way how to test and rank molecules based on different metrics, and this is where our CHEESE embeddings come into play. You can see normally, and most of our competitors used Morgan fingerprint-based similarity search. On these plots, you can see chemical spaces. The left one shows chemical space based on the Morgan fingerprints, and you can see more or less unified and compact chemical space where there is not. It's more or less like single chemical space without any larger islands of structural patterns. On the other hand, if you use our CHEESE embeddings, which are calculated using CHEESE, you can see actually quite distinct structural patterns, as those compounds that are pretty similar to each other based on those embedding, based on the embedding, do create a very separate island on the right part of the plot, and those dissimilar compounds that are actually structurally different from the perspective of the embeddings are in the different island. in this plot, which actually means that our technology using embeddings is able to see some similarities that are not seen by fingerprints approach. And this is the way how we were able actually to distinguish actives versus non-actives from the blind set, because based on the using of embeddings, we could see exact structural differences. In this case, we used shape similarity as aiming on the shape similarities regarding the blind set was crucial for this target, since, as I mentioned, the binding pocket is very narrow and very rigid, so we really needed to follow very strict structural patterns given by those active compounds. And actually, using our CHEESE shape similarity, we were able to retrieve 53% of hits hidden hidden in the blind set. And combining this CHEESE shape similarity search with light GBM approach, which resulted into 33% hit rate alone. When we combined all these two approaches into the consensus scoring and use them, actually our hit rate, our recall was 67%, which was by far the more successful recall of compounds from the blind set from all competitive teams. I think the second best was around 30%. So this is a clear example where the CHEESE embeddings are superior to classical fingerprint-based approach, which was used by the rest of the teams. It's also nicely visible on this plot, where really those compounds that do share some structural similarities are on their separate own island, whereas those another inactives are in their own separate chemical space. And this is the way how we won the first prize at this 9ADD competition a couple of weeks ago. Now I'm approaching the small sneak peek of our most recent technology called SynthonGPT, which is generative model based on actually synthon-based approach of building molecules, where the strategy behind this technology is built on the idea of the deconstructing compounds into single synthons and then finding most closest synthons in the large libraries that we have, and based on those most similar compounds or synthons found in those libraries, combinatorially reconstruct the molecule using different synthons. So in a very short time span, we are able to analyze compound and then reconstruct very similar compounds in a very short time using this synthon-based approach. Our libraries at this point are offering more than 40 trillion molecules in total. Actually, we already have a small project using SynthonGPT technology, using very simple pipeline where we were provided a set of compounds from the docking of the 0th iteration, and the client selected 100 best compounds, which we then expanded using SynthonGPT. And for every single compound for those 100 best, we selected 10,000 closest compounds built by SynthonGPT, and which resulted into 1 million of novel compounds, which were then again docked using the technology of our client, and again selected 100 best compounds. And this process was repeated 3 times in order to get results. Although I cannot show you specific results at this point, I can tell you that results from this iterative approach using SynthonGPT resulted into very interesting compounds with very nice in silico activities. Also, it provided very rich diversity of compounds, and very, let's say, very reasonable structures. So just to conclude let's say, this small sneak peek from the SynthonGPT, the SynthonGPT approach, this synthon-based approach seems to be very complementary to the existing tools that are usually used by our colleagues. It has very high accuracy and very similar numbers of hits compared to very, let's say, similar computational approaches. And we are planning to make some small CHEESE webinar in approximately 3 or 4 weeks. So we will announce this on LinkedIn as well, and everyone is welcome to come and discuss this technology with us. SynthonGPT can be very easily trained for and customized based on the demands of our customer or partner. So we are also eager to hear any proposals of using this SynthonGPT in the projects of our partners. Lastly, I will talk a little bit about CHEESE search integrations, which we are trying to make CHEESE search as easily applicable as possible. And there is multiple way how to implement CHEESE search into the partners or clients pipeline. First way how to implement CHEESE search is via application programming interface API, where we are offering a series of AP calls that are here in the library on our web page. And using this approach, you can programmatically make your pipelines, for example, downloading compounds similar to your input structure, usually SMILES, then downloading, let's say, 1,000 more similar compounds, and then just write the script, for example, which will directly transform your compounds into a docking or another machine learning approach. So there is a quite rich variety of epicals on our website. So if anyone is interested, you are very much welcome to go to use it and explore your chemical spaces using CHEESE search. And also we have a public GitHub repository where we are storing most used scripts, usually downloading most similar compounds to your starting hits, and then using them as CSV for the purposes. So our GitHub repository is also free to use, and everyone is very much welcome to go there and use our scripts. Also, we are offering this full support for the KNIME components. We have a CHEESE search API KNIME component, which can be easily implemented into CHEESE KNIME workflows and used for whatever purposes our client desires to. It's also very easily utilized and used. And also we have a CHEESE search workflow that contains SDF input. So for clients who prefer to use SDFs of their compounds, it's also very easy to use. And lastly, some closing words. Mainly, thank you for your attention. It's been a pleasure to present you our most recent use cases. We are looking for collaboration on every level, both academical and industrial collaboration. So we are looking for partners in this field, and if anyone is interested in some further information regarding our technology, please do not hesitate to contact us. And if you are interested into these slides, also please contact us at info@deepmedchem.com. So thank you for your attention.
Great. Thanks again, Christopher, for the nice presentation. And for the attendees now watching, we are still here to answer your questions. So you can write comments in the LinkedIn post below this webinar or in the chat window. And if you want to ask some direct question or a private message, you can also message us on LinkedIn to our accounts. I see a first question here from Mr. Damilola. And the question is, hello, are we going to see a practical scenario of using the CHEESE embeddings? So maybe, Christophe, you could recap what, how the CHEESE embeddings were used.
Yeah, thank you for the question. I will show you the slides that that are connected to the CHEESE embeddings technology application. So can you hear me, please?
Yes.
Yeah. Because I happened to me that I had some echoes. So actually, when we applied the CHEESE embeddings, was exactly the case of the 9ADD challenge, where we needed to describe the molecule from the perspective not as a structure, but perhaps as a from the perspective of descriptors of that will describe it mathematically as in the n-dimensional space. And for that purpose, because as I mentioned, the docking and classical approaches for the discovery of the hit that or the compound that would bind nicely into this enzyme were actually failing because the nature of the binding cavity of the protein. We needed to describe the molecule from the perspective of this n-dimensional space, which would take in consideration in its space. So that was the moment where those shape-based embeddings came into play, because that was the moment when we actually calculated embeddings of all our molecules, of all molecules from the blind set into these embeddings, and then we compare the values of the set embeddings to each other. So in order to actually gain these plots and actually to be able to compare similarities or cosine similarities in this case of compounds from the blind set, we needed to compare these exact embeddings which you are referring to. So firstly, we needed to calculate shape similarity embeddings, then compare them, and then to create, for example, these plots which are showing the right plot is showing the distribution of compounds that is based on these embeddings. So this is the very specific and concrete example of using our CHEESE shapes in embeddings, where we took into consideration the shape similarity of our compounds based that was calculated or evaluated by the CHEESE embedding techniques. So, and based on that, we were able to actually describe the molecule from the perspective of this n-dimensional space that took into consideration the exact shape of those molecules and then compare them to each other. So I hope this answers your question. This is the exact case where we were applying embeddings in a very practical use case.
Yeah, thanks, Christoph, for the answer. So as you can see on those plots, on the right, embeddings, on the left are fingerprints. And we can see that in these CHEESE embeddings, there is much cleaner separation of those chemical spaces of the blind set compounds versus the actives from Campbell. So that's how the embeddings work, and that shows that they are complementary and see something that fingerprints miss. There are another questions. One question is from Pierre, and that's, hello, thank you for the presentations. Are the computations performed on a cloud, or can it be run locally? I would answer this question. You can run all these computations, specifically this CHEESE search, on cloud, and Christoph also demonstrated the API calls and the 9 nodes, so it's accessible through cloud. But in the on-premises version, which we offer for companies to install CHEESE on their own servers or your own machine, you can run the computations locally, and then you don't have any compute limits, and it's even faster and behind your firewall. So that's the advantage of our on-premises version. And if you are interested, let us know on our email, and we can schedule a meeting and talk about it. The installation is pretty simple, and most companies we work with use this on-premises local version. Then there are other questions. Dr. Chandan Kumar ask if we can share the slides and if there will be a recording. Yes, there will be a recording on YouTube on this session, and I will also send it to your email you posted here, so no worries with that.
Also, if you want us to send you those slides, you can just request the presentation on info@deepmedchem.com. We will happily send you once you provide us email address.
Yeah, we already see the email address in the comments.
I see. So
yeah, I wrote it down, so I will send it to your address. If there are no more questions, this concludes our today's webinar. Thanks a lot again for your attention, Christoph, for your presentation. I'm happy to see you again on our next webinar in a few weeks, which will be focused on SynthonGPT. And if you have any questions in the meantime, you can write us direct message on LinkedIn or send email to info@deepmedchem.com. And feel free also to try our tools on cheese.deepmedchem.com, where you can register for free and try all the functionalities, and we are here to help you. Thanks a lot again, and have a nice day.
So, thank you so much for your attention, and have a great day. See you next time.
Bye.